How vaccines are developed to fight diseases. The majority of vaccines have been in use for decades, and millions of people receive them safely each year. Any vaccine must undergo thorough and rigorous testing to ensure its safety before it can be included in a country's vaccine program. Each vaccine development must first be screened and evaluated to determine which antigen should be used to elicit an immune response. This preclinical process is carried out without using human subjects. An experimental vaccine is first studied in animals to determine its protection and potential for disease prevention. So, what are the components of a vaccine? Vaccines contain tiny fragments of the disease-causing organism, or blueprints for producing the fragments. They also include other ingredients that contribute to the vaccine's safety and efficacy. These latter ingredients are present in the majority of vaccines and have been used in billions of doses of vaccines for decades. Each part of the vaccine serves a particular function, and each ingredient is checked during the manufacturing process. The road to disease prevention, the invention of a new vaccine, is a complicated and time-consuming process that typically takes 10 to 15 years. Vaccines are created using a variety of simple techniques, so let's look at the strengths and limitations of each strategy. But before we begin, support this video and channel by leaving a like right now and smashing the subscribe button. Enjoy! Method number 1. Inactivate the virus. This method uses a chemical to inactivate or kill viruses. Since the virus is killed or inactivated, it can no longer replicate or cause disease. This technique is applied to manufacture inactivated polio, hepatitis A, influenza shot, and rabies vaccines. Since the virus is still seen by the body, cells of the immune system that protect against diseases are generated. This strategy has two advantages. 1. The vaccine cannot cause, not even a mild form of the disease that it prevents. 2. People with compromised immune systems can receive the vaccine. The limitation of this strategy is that it usually takes many doses to achieve immunity. Method number 2. Use part of the virus. Using this method, only a portion of the virus is removed and used as a vaccine. Hepatitis B, shingles, human papillomavirus, and a flu vaccine are all made in this way. The vaccine consists of proteins that reside on the surface of the virus. This strategy can be used when the immune response to a part of the virus or bacteria is responsible for preventing disease. These vaccines can be given to people with weakened immunity and seem to induce long-term immunity after 2 doses. Method number 3. Use part of the bacteria. Some bacteria cause disease by producing a toxic protein known as a toxin. Several vaccines are created by taking toxins and chemically inactivating them. When inactivated, the toxin is referred to as a toxoid. Since the toxin has been made inactive, it no longer causes disease. This is how diphtheria, tetanus, and pertussis vaccines are developed. Another approach for developing a bacterial vaccine is to use a portion of the bacteria's sugar coating or polysaccharide. Immunity to the sugar coating, rather than the entire bacteria, protects against infection by some bacteria. However, since young children's immune systems do not react well to the sugar coating alone, the coating is added to a harmless protein, resulting in a conjugated polysaccharide vaccine. This method is used to manufacture vaccines against Haemophilus influenza type B, pneumococcal infections, and certain meningococcal infections. Like inactivated viral vaccines, bacterial vaccines can be administered to people with compromised immune systems, but they frequently need several doses to induce sufficient immunity. Method number 4. Provide the genetic code for part of the virus. DNA, mRNA, or vectored viruses. Using this technique, the vaccinated individual becomes a part of the virus. Some COVID-19 vaccines are produced in this manner. mRNA vaccines. The COVID-19 messenger RNA vaccine contains mRNA that is the code, or blueprint, for the SARS-CoV-2 virus's spike protein. Dendritic cells in the vaccinated individual use the blueprint to make the spike protein from the virus's surface. When the immune system recognizes a protein as foreign, it mounts an immune response to it, including immunologic memory, so that the immune system can react quickly the next time the individual is exposed to the virus. This technique, similar to vaccine methods that specifically inject parts of a virus, can be used when an immune response to one part of the virus is capable of defending against disease. These vaccines may be given to people with weakened immune systems, but they require 2 doses to be effective. This is how the Pfizer and Moderna COVID-19 vaccines are developed. DNA vaccines. DNA vaccines deliver the genetic code that is used to create mRNA. The mRNA then serves as the blueprint for the viral protein, and the immune system reacts to protect the body and establish immunologic memory by identifying it as foreign. There are currently no commercially available DNA vaccines. Vector virus vaccines. Another way to deliver the gene encoding the coronavirus spike protein is to put the gene into a virus that cannot replicate itself, but can still enter the cell and deliver the desired gene. This strategy is used in so-called replication-deficient human, or simian adenovirus vaccines. Although adenovirus can cause human disease, these vector viruses are carefully designed so that they do not cause disease. Therefore, they can be provided to people with weakened immune function. The Johnson & Johnson COVID-19 vaccine is made in this way. It is given in 1 dose. If you like this video and would like to hear more, be sure to press the subscribe button.